Thank you so much, Kat, and thank you for inviting me to talk today. I am really excited to share this project with you. And it's really great to connect with you guys and see what you think. The project is quite new. And as the title suggests, what we're trying to do is to look at the role of cultural innovation in how diseases actually emerge. So we have obviously a large set of data, especially since the emergence of COVID, and also a lot of modelling that's been done when diseases have emerged, how they have progressed due to our behaviour, the way that we change our practices, whether we wear masks or don't wear masks, whether we segregate in areas, whether we stay divided. So all those things have been studied. But what we would like to do is take a step back from this and think about how our behaviors or how our changing, how cultural changes and innovations can actually give rise to pathogens and their emergence. So a little bit about the link between cultural innovation and the emergence of new diseases. So when we partake in some sort of a new cultural practice, or there's some sort of a new innovation, or perhaps we just start to eat something new that we found in the Amazon. What we start to do is we start to have this amplified interaction between humans and microbes. Some of these microbes might be something that we already have interacted previously, or there might be new microbes. And what this interaction can do is it can amplify the microbe populations. And through this interaction and this amplification, we have a higher risk of a possible mutation occurring. And the mutation can then give rise to a new pathogen. Now, there are many instances in which our activities have given rise to new pathogens or the re-emergence of pathogens, and these include HIV, Ebola, SARS, and COVID, along with others. So I want to give a couple of examples of distinct examples of when we think that emergence has been caused by cultural innovation. So the Nipah virus is thought to have originally emerged due to deforestation to make room for agriculture. One theory is that the forest areas were cleared to make room for commercial fruit production. The native bats in that area carrying the virus were eating the fruit and dropping some of the contaminated fruit on the forest ground. And, there was pigs in the pig farms in that area. They ate the fruit, and then the disease was passed on to humans. There's another theory that the deforestation and burning of forest land disturbed the natural habitat of the bats, forcing them to move closer into farming areas. In either case, we can see that how the change in the microbial environment has driven mutations and pathogen emergence. The other example I'd like to talk about is legionnaires disease. So in 1976, there was an outbreak of a severe type of pneumonia amongst the delegates at a conference in Philadelphia. This bacteria, which caused this type of pneumonia, was later known to be Legionella. Now, Legionella bacteria is actually found in natural water reserves. The building of cooling towers for air conditioning systems actually amplifies this bacteria and caused the emergence of Legionella disease. So here are a couple of examples of how cultural behaviours, cultural innovations have given rise to a pathogen. So what are we trying to do in these projects? Well, there is our overarching aim is that we want to understand how new diseases emerge when we have cultural shifts. What is it about these changes that causes emergence? What is it about our behaviour? And what conditions are there that induce emergence? And if we understand these conditions and we understand our behaviours that are causing emergence, can we somehow predict or mitigate these conditions? So I want to introduce you to the initial model. So this is the model that we're going to use to build to build future work on. And this is quite a general model of cultural practices and emergence of a pathogen. So here we've got a population. Let me just describe the model using my pictures. Here we've got a population of people. They're living in a community, and all of a sudden, one of them has this great idea. And through social interaction, this person starts to tell other people about this great idea, and other people begin to choose to kind of partake in this new practice. But by partaking in this new practice, people then begin to interact with some sort of a microbial population, and the microbial population begins to amplify. And then at some stage, we end up with a mutation into pathogens. So this is the overall idea of the model. I should also mention that the people partaking in this new activity, the people in the red, can revert back. They can abandon the activity. So let's take a look at the deterministic processes in this model. So we built the initial model as a system of ODEs, and I want to describe the ODEs, and then I'll move it into the stochastic model. So here we've got the initial population, the N1 population. Through social learning, they move into the N2 population. So I really need to stress that this is all within one community. These people are not separated anyway. And in the ODE system, I mean, they're just well-mixed populations. So essentially, these people move into taking on this practice. The practice can also be abandoned, and the population moves back into N1. This is death due to interaction with the actual pathogen. And we have this growth term for the people who take part in this activity. So there's some kind of fitness advantage or reproductive advantage for taking on this new activity. Now, the pathogen population grows due to its interaction with N2. This is a very general term. It just describes a proportional growth as N2 interacts. It's a very general growth term, which is just proportional to the amount of N2 people that are around. There's a death term for the micropopulation, and the microbes mutate. The population of pathogens, they grow due to interaction N1 and P, N1, N2, and P. We've got the death rate again, and we have this mutation. Now, we have some initial conditions for the model. Let's assume that initially we have a large population of people within this community. So that, let's say there's 10,000 people. There is one person who partakes in the activity, and there's no pathogens or mutants. There are no microbes or pathogens in the environment. For anyone who's interested in how the model is structured, I need to explain that 1 unit of microbe or pathogen is actually not the same as 1 unit of the humans. So you can imagine that 1 unit of microbe or pathogen equates to an average amount of microbes that you may find around a person of a particular strain. So that's the deterministic model, but the mutations in this model are a rare event, and we want to describe these rare events and see what happens as the populations progress, as opposed to having this mutation term which just runs from one population to the other. So to do this, we transform the model into a stochastic process, and these are just the propensities for the changes in the populations, and we run this as a exact Gillespie method. Okay, so let's take a look. Actually, before we take a look at the results, let me describe what interactions we're actually interested in this model. Well, we're interested in how human behavior affects pathogens. So there are a few things that we're interested in studying here and seeing what the result is on the emergence of the pathogen population. So the first thing we're interested in is the speed at which people adopt or abandon the new practice. The other thing that we're interested in is whether the reproductive advantage has any effect on the emergence of the pathogen. So they're the things that we're going to investigate in this model. These slides are quite busy, so I will explain as we go. So the first thing we're going to look at here is the effect of social learning. I've run these models for 350 years. So this is 350 years of running the models. And what we've done here is we stop the simulations at the point of emergence. And the reason we do that is this model can't describe anything beyond emergence. As we know that there's a lot of human interventions that occur after a disease emerges. There is medical interventions. So all we're interested in is emergence. So beyond emergence, we stop the model, and we say emergence is around about P of 100. So we said 100 units of the pathogen. So what happens in this model is if we look at one of these realizations, we can see that the N1 population starts to adopt the new practice, and then the N2 population takes over. There is the N1 population will reach some sort of a steady state due to abandonment. And as a result of the N2 population growing, the micropopulation grows, and at some stage we get a pathogen population. Now, when we have fast social learning, this process occurs a lot sooner. So this transition occurs sooner. The micropopulation grows faster emergence. If social learning is slow, well, we have this slower transition point. There may be a few times where the pathogen is going to try and emerge. And in the end, once the M population is big enough, then it's going to emerge. So remembering that the M population grows proportionally to N2. So essentially, in this very simple model, if N2 gets big, then the micropopulation is going to get big, and hence the pathogen has more of a chance of a microbe has more of a chance of mutating to a pathogen and surviving. Okay, so here what I've done is simulated the model 200 times for each of these social learning rates. So what we see is that as we increase the slow adoption of the practice with low abandonment delays pathogen emergence. And emergence is almost certain in all cases. What I mean by this is that if you run this model long enough, well, if the N2 population continues to grow, eventually we're going to get emergence. This model, again, has been run for 350 years. Okay, so let's take a look at what happens if we think about abandonment. So here we've got low abandonment, so we can see that the N1 population reaches a steady state somewhere around here, and the N1 population in high abandonment case is much larger. So faster growth for N2 here, faster microbe emergence, and earlier time point to pathogen population emerging. And in this case, again, we have this kind of slower uptake. And there's some cases where, the pathogen has tried to emerge and eventually it's emerged over here. So abandonment delays pathogen emergence, which is, logical, and the probability of emergence. So we have this changing probability going from 86% to 20%. And as I said, we have this delay. But again, depending on how large I make this abandonment, most likely, eventually, the pathogen will emerge, be it 800 years down the track. But then there's other cases to take into consideration. Will people change their practices? What could happen in the meantime? Which is where we take the model next. So let's extend this model a little bit to look at a case where the population comes up with a safer version of this practice. So far we've got N1 and N2, and then this guy is like, I have this great way to do this thing. And so we have this social learning again between these two populations, between N2 and N3. And at this stage, there is no abandonment from here. So, imagine that once you've committed to this, the rate of abandonment is so low. Although I will talk about that at the end. But essentially, let's pretend that these populations move into this phase, and if they move into here, they're not going to abandon. They also are getting the benefit from this population, and the N3 population can also die once a pathogen emerges. The initial conditions in this case is that it's that both N2 and N3 are at 1 initially. There's other ways that this can be done, but let's assume that, there's two people who come up with this idea around the same time, and one's a bit safer than the other. Okay, so what do we see now when we look at the results from this model? So this is the social learning effect of N2 to N3. So as we increase the rate at which people practice this new method, there's a clear reduction. Oh no, I'm sorry, I've taken that out. We have a clear reduction in the probability of emergence. Sorry, I didn't put that one in. So probability of emergence in this case moves from 0.68, so 68%, to nearly about 1%. And that makes sense because what's happening in this case, and I haven't put this result here, but I will show you in the next slide. When you have fast social learning, we really reduce this window in which the N2 population is practicing. So as we reduce this window, there's less chance of the microbe population taking over and hence the pathogen emerging. Slower social learning, however, leads to really variable results. And this can occur for a few reasons. One of them being the abandonment of practice. The other one is also just from the pathogen emerging early and sort of killing off this population early on. How realistic is that? I don't know. So, but it's just an artifact of the model as well. Okay, so so the idea there is that if social learning happens fast enough, we can actually completely avoid emergence. Because in this case, what happens is the N2 population isn't around for long enough to cause emergence, and so we can totally bypass emergence or make the possibility really, really small. One last slide that I want to show for this model is just to take a closer look at abandonment and fitness advantage. So this is the results from the actual ODE system with no pathogen. So essentially what we have here is if we have, this is no abandonment, this is relatively small abandonment, moderate abandonment, and this is large abandonment. So if we look, for example, between this case with the moderate abandonment and large abandonment, we see is that the N2 population, the period of time which this population is around is actually increasing. So although the total number of N2 population is smaller in these cases, in this case, as opposed to the moderate case, the period of time that these people are around is actually longer. And so so the question there is, if there's anything that can drive the N2 population to also grow, such as the growth term itself, so if they have a higher fitness benefit, or if there is abandonment between N2 and N3, then we can still drive emergence. And so what I did was I just had a look at the probability of emergence from the Gillespie algorithm for a 200-year time frame to see if we see something like that. Well, if we compare a reproductive advantage to abandonment, there's a clear trend that as we reduce abandonment and increase reproductive advantage, we're going to increase probability of emergence. But if also we just move from this low reproductive to high reproductive advantage, we can see that there's also that the probability of emergence can also increase. So yeah, that's another interesting dynamics that occurs within this relatively simple system. Okay, so what I want to do now is just to summarize the results from this preliminary model. So what we've seen is that the speed at which we adopt new practices can increase or reduce the risk of emergence. without a safer option it's for the population to partake in, emergence is delayed, but it is pretty unavoidable if no other changes are made. And as I said, that's a really broad statement, but we're just looking at a very simple model here. If a safer option is embraced quickly, then emergence can be totally avoidable. Abandonment of the practice and fitness advantage are also important factors driving emergence, as we just saw, and they require further investigation. And then importantly, there's a couple of questions here that I'm thinking about, which is abandonment may result in a reduced risk of emergence, but what does that mean for progress? And, are we seeing a trade-off between advancements and management of pathogen emergence, which is, I think, that the more important question here. So as for future work with this model, what I've shown you, as I said, is the preliminary initial model that we've been investigating. What I'm looking at is extending the model to include more complex behaviours, such as conformity within the populations, moving from a single mutation process to multiple mutations. So, you have a few mutations before you reach this pathogen state. A really important thing that I haven't shown in this model, which I am actually investigating right now, is different ways in which the microbes exist and amplify in the human population. So at this stage, the amplification of the microbe is very generally just linked as proportional to N2 population. But, this is obviously a more complex mechanisms that drive this, that drive the amplification. We're looking at extending the model to include infected and recovered populations. So this is work that Yasmin Oren is actually undertaking right now. She's our honours student. And what she's looking at is a shorter window and studying an SIS model and looking at other variations, such as humans just interacting with a wild animal or with a domesticated animal. So hopefully in future I can show you some of that work as well. Now, importantly, we've shown a sort of a proof of concept model here, but how are we going to calibrate this model to study diseases of interest? And this leads me to the second part of the talk that I would like to share with you. Our team is also looking at clustering diseases based on their emergent variables. So the project aims to identify classes of diseases based on factors that have driven their emergence. So this is a project that I have, I'm showing on behalf of the group that I've actually worked on it. So Sarah Liu, who was Mark's previous postdoc student, really drove this project along with Jeremy Kendall, who is our collaborator from Durham University, Mark Tanaka, my supervisor, and Catherine Marley. So I'm hoping that I can share this project with you and show you how we're planning to inform our mathematical model. So the team came up with the research questions, what are the known or proposed origins of infections of diseases, and what human factors contribute to their emergence or sustained transmission? So the team did a large scoping review and identified 58 diseases of interest and 26 emergence variables. The diseases of interest were highlighted as those that are of current public health interest and those that have a large prevalence in human history. So let's talk about the emergence factors, the emergence variables that were identified. So these come in two classes: human factors of emergence, so things such as proximity to animals, animal domestication, movement of people, poverty, urbanization, all those factors. 15 in total, which are to do with how humans may be involved in the emergence. And then we have modes of transmission, so direct contact, fluids, sexual, vertical, vector-borne, and 11 of those in total were identified. So this is where I'm hoping that my wheels aren't going to fall off. So please bear with me as I am relatively new to this type of statistical analysis. So what the team did was they transformed the survey data into a binary matrix. So the presence of a factor was deemed was noted as a 1, and the absence was noted as a 0. Now, the absence here is either due to a factor not being present or the lack of information from literature. So in this case, what they did was they used they looked at similarities between diseases using a Jacquard similarity index. And then diseases were clustered using the UPGMA method, and they found the optimal number of clusters using Radkowski-Lantz index, and they found that to be 6. So the diseases were clustered in this way. And as I said, this is a very exploratory initial idea here, which I thought would be very interesting to share with you. So they then did a non-metric multidimensional scaling, and this was performed for visualization of clusters. I know there are people here who are much more familiar with this, and they found a stress factor of 23.96. So the stress factor is higher than desired, but as I said, we're exploring how these diseases are connected. So we can see that, they're not, the groups aren't very, very distinct. However, we continue. So the team characterized the clusters by looking at the distribution of the presence of emergence variables in each cluster. So this is what I'm very interested in when it comes to informing my model. So we have 6 clusters here. The first cluster is the environmental contamination cluster. We have these human-animal interface clusters. And then down here, there are 2 clusters which only include one disease. So we got Legionella and pertussis. So I just want to talk a little bit, talk a little bit about a couple of the clusters here. So for the environmental contamination cluster, these diseases tend to be environmentally transmitted. Emergence factors that increase water contamination or contamination of living environments appear to be driving the mechanisms of emergence. These include poverty and low socioeconomic conditions. Most of the helminths, I think they are the worms. The worm diseases are clustered in this category as their emergence is dependent on water quality. I really hope I'm not butchering their work, and I apologize if I am. So what about cluster 2? So cluster 2, as another example, is human-animal interface cluster. Emergence of these diseases is likely to be tied to contact with an animal reservoir. Transmission is facilitated by proximity to animals and the movement of animals. Emergence is likely to be related to increased contact at the human-animal interface. These diseases are actually well studied and thus have a wide range of factors wide range of factors and diseases. So we can see that because they're better studied, we have a lot of interesting factors in there. Legionella is an interesting one because the information is really limited on this disease. As I said earlier, it is related to a specific human innovation, which is air conditioning. So yeah, it happens to be clustered on its own. Pertussis is very similar. There is a single cluster separated from the other diseases. Information regarding the origins of this disease are unclear. So it is important. So it is distant from other diseases. Okay, so as I said, the important idea here and and what I really wanted people to take home is that this project and the initial results from this project can inform the mathematical model and give us an idea of the type of behaviors that are causing particular diseases to arise. Okay, so as I fumbled my way through that part of the presentation, I would just like to give an overall summary of this. We have identified clusters of diseases based on similarities of emergence variables. Although a signal of clustering is present, this signal isn't strong. And one reason for this is that the data is sparse for particular for particular diseases. We're not putting any weight on the zeros in the data, and this could be problematic because some of the zeros, as I mentioned, is because the disease there's been no that we don't think that the disease emerges due to these factors. However, there are zeros which are there because we don't know, or there's not enough information. So this makes it a little bit more difficult. However, the patterns that emerge in this study allow us to further investigate the emergence of diseases based on cultural and environmental drivers. I would just like to thank and acknowledge the team, Mark Tanaka, my supervisor, Sarah Liu, who's done a lot of this work. And I'm sorry, Sarah, if you ever listen to this, I did my best. Jeremy Kendall, who we work with from Durham University, and Yasmin Oren, who is our honour student. Thank you very much for your time today. I appreciate it.